Hi everyone, welcome back to Dr. Han's Immunology series. Now today we are back to talk about how our bodies defend against infections and pathogens specific to the mucosal surface. Now previously we have already briefly talked about the barriers and IgA, and in this lecture we will expand more on the specific details. So let's get started. Here are the lecture objectives. We will look at how mucosal barriers and innate and adaptive immunity interact with each other and play a role in mucosal surface immune responses. We will also compare and contrast the differences between systemic and mucosal immune responses. And lastly, we will wrap up with how we eliminate gut parasitic infections. When we look at the mucosal tissues in the body, because of their physiological function, they require communication with the environment, and mucosal surfaces are by necessity dynamic, thin, and permeable barriers to the interior of the body. For example, lungs mucosal for gas exchange and gut mucosal for food absorption. But these features also make them more vulnerable to infection, invasion, colonization. So the body has evolved multiple mechanisms to immune defense, including barriers, innate, and adaptive responses. Let's first look at various barriers on mucosal surface that provide the first line of defense. Mucin is a negatively charged glycoprotein that is produced and secreted by Goblet cells on the surface of the epithelium. It is an example of a mechanical barrier that traps bacteria and works best with ciliary action to remove bacteria from various mucosal surfaces. Epithelial cells are arranged with different specific junctions to prevent both commensal and pathogenic bacteria from invading internal tissues. The first one is the tight junction. It is a type of impermeable junction that prevents molecules from passing through intracellular spaces. Second, a desmosome. It is a network of fibers that bind adjacent cells together. To visualize this type of junction, you could imagine it is a cellular Velcro. The third one is gap junctions. The name suggests it has the widest space between the adjacent cells. This type of junction allows communications between two cells via ion passing through the channels. Microbial populations may be found on the skin, in the lower respiratory tract, mouth, and lower GI. Throughout life, these resident organisms vary in type and number, and individuals can have significant differences in their resident populations. Commensal microorganisms provide a microbiological barrier of defense. They are mutual organisms that use the host for shelter, food, and support and transportation. At the same time, the host also receives benefits in digestion, protection from infection, and clearer skin. So we call this a symbiotic relationship. The commensal organisms are therefore benefit to us. The GI tract contains the most amount of commensal microorganisms, in part due to very high surface area. And they have many functions in the gut. For one, they are responsible for synthesizing vitamin K, which is a cofactor for blood clotting. Some drugs also rely on commensal bacteria to convert them into active metabolites. For example, a drug called sulfasalazine is converted into active 5-ASA mesalamine by colonic bacteria. Another very important function of commensal bacteria is their ability to degrade plant fibers into short-chain fatty acids, such as butyrate. Studies have shown butyrate plays an important role in many aspects of health, such as weight management and blood sugar levels. These bacteria can also help degrade toxins found in food, such as mercury in fish and hydrocarbons found in grilled food, into harmless molecules that can be either excreted or used in other physiological functions. Commensal bacteria colonize the gut and occupies the spaces and compete for resources with disease-causing pathogens. Their constant presence also triggers T-cell presence and development in the secondary lymphoid tissues present in the small intestine. So now let's look at the secondary lymphoid tissues in the gut, namely the gut-associated lymphoid tissues, or GOLT, mesenteric lymph nodes, and Peyer's patches. GI tract is defended by various secondary lymphoid tissues. GOLT monitors the gut lumen and initiates immune response when there is pathogenic invaders. Both innate and adaptive branches immune cells are present in GOLT, namely B cells, plasma cells, T cells, macrophages, and eosinophils. The mesenteric lymph nodes are gut-specific lymph nodes that line and protect the gut. Invading pathogens in the gut are taken up by these lymph nodes. And third, we have Peyer's patches. M cells are specialized cells in the Peyer's patches that provide a small window to uptake pathogens. Peyer's patches also contain germinal centers for B cells and T cells development and activations. The most notable features in the gut is that the immune response is different than the systemic response, and we will explore how and why in the next few slides. Here we have the anatomical organization of the GOLT. Now notice Peyer's patches are only found in the small intestine, and M cells are embedded within the epithelium and create an opening for pathogen uptake. Another notable structure is the isolated lymphoid follicles. They are found in both small and large intestine and are particularly concentrated at the appendix. Only B cells are found in these isolated lymphoid follicles, in contrast to having both B cells and T cells in the Peyer's patches. This is an illustration of the mesenteric lymph nodes. B and T cells activated in the Peyer's patches leave the lymph and travel via mesenteric lymph nodes and the thoracic duct to the blood. Now, once back in the bloodstream, these mucosal activated B and T cells travel back to the type of mucosa in which they were activated and entered it from the blood. This means the activated B and T cells would go back to the intestinal mucosal and mount an immune response. Before we talk about the mucosal immune response, let's have a quick review of the systematic immune response. For example, when there is a cut, pathogens invade tissues. We have macrophages and dendritic cells come in and eat up the pathogen. Then they release pro-inflammatory mediators that recruit more immune cells to the site of infection, leading to local acute inflammation. Now, if it is a cut, blood clot would also form, so inflammation is the hallmark. But in contrast, the constant presence of commensal microorganisms in the intestinal lumen epithelium does not cause inflammation. Even when commensal bacteria are endocytosed and gain access to the lamina propria, there is still no inflammation under healthy conditions. This is because effector B and T cells and IgA are there to maintain the balance and prevent commensal bacteria from invading deeper into the intestinal tissues. So the key here is that there is no inflammation in mucosal immune response. The reason for no inflammation is that intestinal macrophages are different than macrophages found in other tissues. These intestinal macrophages still eat and kill mucosal pathogens, but they do not produce pro-inflammatory cytokines and lack the co-stimulatory molecule B7, so they cannot act as an antigen-presenting cell to activate naive T cells. They also lack crucial cell adhesion receptors such as Fc receptors, LPS, and express very few toll-like receptors, so they do not activate the NF-kappa B pathway that leads to the inflammatory response. Dendritic cells reside in the gut also play a special role. They are particularly important for maintaining oral tolerance. These immature dendritic cells can extend processes like an arm that reaching across the epithelial layer to capture food antigens in the lumen. Even though these dendritic cells present food antigen to naive T cells in the mesenteric lymph nodes, they lack co-stimulatory molecules to induce T cell activation into Th1 or Th2 helper cells. Instead, these naive T cells differentiate into regulatory T cells that secrete IL-10 to suppress immune responses to food antigens. These regulatory T cells are abbreviated as Treg in most textbooks. They belong to a subtype of CD4 T cells. They secrete IL-10 that suppresses macrophage activation and decrease Th1 cell activation. They also release TGF-beta to decrease B cell activation. Treg cells are particularly important in maintaining a non-inflammatory intestinal condition. We have observed that inflammatory bowel disease patients often lack Treg cells in their gut and have overactive T helper cells, which lead to autoimmune diseases such as Crohn's disease or ulcerative colitis, where are the two types of inflammatory bowel diseases. In the figures, the light micrograph is a section of gut epithelium and shows villi and a Peyer's patch. The T cell area and a germinal center GC are indicated. It appears as a sunken area of the epithelium that has characteristic microflows or ruffles on the surface. M cells capture microorganisms from the gut lumen and deliver them to the Peyer's patches and the lymphoid follicle that underlie the M cells on the basolateral side of the epithelium. M cells are specialized for the uptake of pathogens from the intestinal lumen and deliver them to the basolateral side of the M cell membrane. Now here, dendritic cells and B cells take up antigen and stimulate the proliferation and differentiations of antigen-specific T cells. After antigen presentation, activated T helper cells stimulate B cells to become plasma cells. These mucosal plasma cells preferentially secrete dimeric IgA that is specific for pathogens, commensal organisms, and food antigens as well. The goal here is to maintain a balance of all these antigens and prevent inflammation from happening. B cells and T cells are first activated in the Peyer's patches. Then they migrate to the mesenteric lymph node and differentiate. Following differentiation, these mucosal-specific lymphocytes, or we call them gut-homing lymphocytes, contain specific adhesion molecules that allow them to travel through the bloodstream and the lymph and back to the intestinal mucosal tissue. These specific adhesion molecules are integrin alpha-4 beta-7 and L-selectin. They are expressed on both gut-homing B and T cells. These adhesion molecules bind to mucosal vascular adhesion, or MAdCAM1, that is on endothelial cells of blood vessels in the gut wall. The binding between MAdCAM1, integrin, and L-selectin allows lymphocytes to leave the blood vessels and enter the lamina propria. In the lamina propria, gut-homing lymphocytes use CCR9 chemokine receptor to bind to CCL25 chemokines secreted by the gut epithelial cells. These features allowed gut-homing lymphocytes to go back to the intestinal area and perform their functions. Gut-homing B cells differentiate into plasma cells and undergo isotype switching to primarily secrete the dimeric IgA antibody isotype. This IgA is transported to the mucosal surface and perform neutralization and opsonization. Before we move to the final section of this lecture, let's look at this riddle. I can swim, crawl, or fly, but occupy neither sea, earth, nor sky. I'm a most uninvited company, taking without giving a thing. What am I? Well, the answer to the riddle is parasites. Parasitic infections are not a significant problem in any industrialized countries. However, for those of us who have cats and dogs at home, we do have to deal with it or prevent worm infection from time to time. Parasitic worm infections are typically located in the mucosal tissues. Helminths are parasitic worms that live and reproduce in the intestine, and they are not commensal organisms. They compete with the host for nutrients and cause damage to intestinal epithelial and blood vessels. Beyond the intestine, they can also reside in other organs such as liver, muscles, and lungs. During a parasitic worm infection, dendritic cells take up parasitic PAMs or PAMPs and activate naive T cells. Th2 helper cells play a crucial role in producing an immune response to parasitic worms. They produce IL-5, which recruit mast cells and eosinophils to parasitic infected tissues and activate them to release granules to kill the worms. Th2 also drives B cell activation to produce more parasitic-specific IgE that lead to neutralization and elimination of worm. These IgE can also bind to Fc epsilon receptors on mast cells and cause them to further degranulate. Now, talking about IgE, we have a dedicated lecture on how IgE plays a role in type 1 hypersensitivity and how it is believed that parasitic infections are tied to lower incidence of allergic reactions. So if you want to learn more, please check out that lecture as well. That is all for this lecture, and I will see you next time. Bye.